And welcome to our video lecture on topic 6.5, neurons and synapses. Our essential understanding for today: neurons transmit the message and then synapses modulate the message. We're going to talk a lot about what neurons are and what synapses are in this lecture. So our objectives for today: we're going to describe the structure of a neuron, we're going to outline saltatory conduction of impulses, we're going to explain propagation of the resting potential, and then we're going to explain the stages of an action potential. We're going to annotate an action potential graph and then define that thing we call a synapse. We're going to describe the stages of neurotransmission and then outline the impact of these neonicotinoid pesticides on neurotransmission. First up, let's talk about some neuron structure. Neurons, also known as nerve cells, also sometimes called neuronal cells. Their job is to transmit electrical impulses, and they do that using ions that set up electrochemical gradients. The stars of electrochemical gradients in nerve impulses are potassium ions and sodium ions. We're going to talk a lot about that coming up. In us humans, we've got about 85 billion neurons in our brains alone, 46 miles of neurons around our whole entire body. Some of those nerve cells are crazy long. The sciatic nerve goes from our feet all the way up into our spinal cord. Crazy long. The reason that we can send impulses really fast from our feet all the way up into our spinal cord in our brain is because nerve transmission is saltatory. This is sometimes also pronounced saltatory. Saltatory conduction is based on the amazing structure of nerve cells. We're going to talk about that structure now. So nerve cells have lots of the same pieces and parts that a normal cell will have. There's cytoplasm in the cell body. We call the cell body of a nerve cell the soma. There's a nucleus, there are ribosomes and endoplasmic reticulum and all those super cool things that cells have. And there are some special things that are unique to nerve cells. One of those is a dendrite. So all of these branches are dendrites, and these are going to collect information from the surroundings of this nerve cell. Often it's going to be neurotransmitters that are released from other nerve cells. We're going to send out a little projection from the soma, from the cell body, and that's called the axon. So this crazy long branch is the axon of the nerve cell. That axon is going to end in axon terminals. Sometimes we also call this a motor end plate, if this happens to be a nerve cell that talks to muscle. Wrapped around the axon is myelin sheath. And this myelin sheath is composed of special kinds of cells called Schwann cells. And Schwann cells are super lipidy. So we've got lots of lipids in these Schwann cells, and these act just like an insulator around a copper wire. And so what happens is all of the stuff that we're going to be talking about, the nerve impulse details that are coming up, they only happen between the Schwann cells. So all of the stuff that we're going to talk about, and you're like, what are we talking about even? When we talk about the nerve impulse, none of it happens here. It only happens between the Schwann cells in these special little divots that we call nodes of Ranvier, sometimes also called nodes of Ranvier, nodes of Ranvier. And so our impulse will come from the dendrites, and then it'll jump between the Schwann cells from one node of Ranvier to the next because of that insulating effect of those Schwann cells, the myelin sheath. If you are in Spanish, you know that saltar means to jump. And saltatory conduction jumps from one node of Ranvier to the next until it gets to the very end of the nerve cell, the motor end plate or the axon terminal. And then here, neurotransmitters or some other kind of chemical signal will be released and sent out to whatever is next. So let's practice that information super quick. This diagram shows a motor neuron. Motor neurons are nerve cells that talk to muscles and make things move, hence the motor. What are the biochemical nature and function of the myelin sheaths? We just talked about that myelin sheath here, composed of Schwann cells. Between those Schwann cells are our nodes of Ranvier or Ranvier. Axon is here, axon terminals. We've got our cell body, the soma, and then dendrites are here. The axon is under through the myelin sheath. So biochemical nature of the myelin sheath, it is very lipidy. Lots and lots of fats, which is not going to provide an energy source, but instead is going to allow for saltatory jumping conduction. The nerve impulse will come from the dendrites along the cell body, and then it'll jump, jump, jump between the Schwann cells from one node of Ranvier to the next until it gets to the axon terminal. Cool beans, right? So amazing. So before we can send out those saltatory nerve impulses, we need to achieve resting potential in our cells first. Resting potential is an electrochemical gradient equal to about negative 70 millivolts. How this happens is we have these amazing pumps. Remember that anytime you see pump, you're going to know that these are transmembrane proteins, and they're going to use ATP. We have some active transport going on. If we saw the word channel, then we would not think active transport, we'd think passive, but a pump is going to imply some active transport, some ATP required. What these pumps do, these sodium-potassium pumps, sodium ion, potassium ion. So sodium-potassium ion pumps are going to pump 3 sodiums out. 3 sodiums get pumped out, and 2 potassiums get pumped in. The sodium-potassium pump is going to use ATP to pump, to push with ATP 3 sodiums out for every 2 potassiums that come in. 3 sodiums out, 2 potassiums in. Again, 3 sodiums out and 2 potassiums in. 3 sodiums out, 2 potassiums in. We're going to use that pump. 3 sodium ions get pumped out of the cell and into the surrounding space. 2 potassiums get pumped into the cell, into the cytoplasm. We use ATP to make that happen. That results in what we call a gradient, an electrochemical gradient, because we have different concentrations of these ions inside and outside of the cell. We have more positive stuff outside the cell, which means that inside, compared to outside, is a little bit negative. It's just about negative 70 millivolts for most cells, and that is what we call the resting potential. Here are all the words that we just talked about. If you want to pause the video and read, you can. Otherwise, let's talk about what happens next. Once we have that resting potential achieved, we can move into an action potential or a nerve impulse. So something will stimulate the nerve cell, and then depolarization can happen. Depolarization is where we flip the inside from negative to positive. So the cell goes from a negative 70 millivolts to positive, maybe about positive 30 or 40 millivolts, because we let all that sodium in. So depolarization, we're going to go from negative to positive. So all that sodium that we worked so hard to pump out with the pump, what we're going to do is take this sodium ion gate, which was closed, and we're going to open the gate, and then all that sodium that's highly concentrated outside the cell can get into the nerve cell. We used to have way more positive stuff outside and fewer positive ions inside the cell, but now look, so many positive ions inside the cell. We're up to like positive 30, positive 40 millivolts. This is known as depolarization. We're going to open up the sodium channel. Sodium can flow into the cell. We call that influx. And that's going to flip the cell from being a negative 70 millivolts to maybe a positive 30 millivolts. Good? That flip from negative 70 millivolts to positive 30 millivolts is going to trigger the opening of another channel, more facilitated diffusion. This time the potassium channel is going to open. All that potassium that we worked so hard to pump into the cell is going to flow out of the cell, which now, notice, we used to have way more positive things inside the cell. All that potassium got out, and now there's more positive outside the cell, which means that we flipped the inside of the cell back to negative, which means we're going to call this repolarization. So we had set up that pole of positive outside to negative inside. In depolarization, we let the sodium in, which flipped it from positive to negative outside and positive inside. But now we're going to repolarize the cell, and we're going to go back to positive outside and negative inside. And we do that again by opening the potassium gate, which is going to allow the potassium to flow out. We call that efflux. Potassium flows out. And then we have repolarization. We go back to negative. What's super cool is that all of these things can be happening all at the same time along the whole entire cell. So stimulus hits here. This little piece of the axon will be depolarized. This depolarizing triggers the depolarizing of the next piece of the cell. As this one gets depolarized, this one gets repolarized. This one is going to trigger the depolarization of the next piece of the axon. Repolarization will follow it. And then that sodium-potassium pump is going to get back to work and get us back to resting potential. And this only happens between the myelin sheath. So those Schwann cells, remember, are going to insulate the axon. So all of this depolarization, repolarization, resting potential only has to happen at those nodes of Ranvier, because our impulse is saltatory-saltatory. Cool beans! How do we know this? So we know this because of this amazing woman, Ida Henrietta Hyde, and she invented the microelectrode. We literally stick tiny little electrodes into the axon, keep one outside the axon, and then we can use an oscilloscope to measure those electrochemical differences inside and outside the cell. So amazing. Let's talk about that graph that we can build from the oscilloscope. So this is showing us an action potential. This graph is showing us a nerve impulse. We start here at just about negative 70 millivolts. That's our resting potential because of our sodium-potassium pump, pushing 3 sodiums out and 2 potassiums in. When an impulse, some kind of stimulation, hits the nerve cell, we're going to get up to just about negative 55 millivolts. We're going to call that threshold potential. I'll talk about that more on the next slide. And that's going to trigger the opening of the sodium ion channels. So sodium ion channels will open, and all that sodium that we pushed out is going to flow in. I have more positive things inside the cell, and that's going to cause the charge inside the cell to get super positive, maybe up to like a positive 30 millivolts. This change from negative to positive is known as depolarization. And it's going to trigger the opening of the potassium gates. Potassium-gated ion channel is going to open. That means that all the potassium that we worked so hard to get inside the cell is going to get out. We call that efflux. Because the potassium is leaving the cell, there are fewer positive things inside the cell, and so the cell's charge is going to get negative because the potassium is going out. We call that repolarization because we're going back. Whoa, polarization. Sorry, spelling can be hard. Repolarization because we're going back to negative. Depolarization, we go from negative to positive. Repolarization, we go from negative back to positive. And then we have this little dip-de-doo that we're going to call hyperpolarization, or we can also call it the refractory period. And this is because that potassium is still getting out, but now the pumps are starting to engage, and we're also pushing the sodium out, and so we get a little bit of extra negative here, hyperpolarization. And then everything, those gates close, and the pump gets back to work. And then we can go back to resting potential and do the whole thing all over again. Here's another look at that impulse graph. This time we're going to talk a little bit more about that threshold potential. So here we are at negative 70 millivolts resting potential. We are ready to send a signal. But here's the thing, our bodies are being bombarded by stimuli all the time. We don't necessarily need to know about every single stimulus ever. There are carbon dioxide molecules and oxygen molecules and nitrogen gas molecules flying into your face right now. We don't need to know every single time a nitrogen gas molecule hits your nose. And so some stimuli are so small that they don't actually set off a whole entire impulse. For us to have the whole entire depolarization, repolarization process occur, we need to have a stimulus that gets us at least to that threshold potential. Threshold potential is just about negative 55 millivolts. We don't need an impulse for every single stimulus ever. And so we need to get a stimulus that is at least big enough to overcome that threshold potential before the whole entire thing happens. If we do max out that threshold potential, then depolarization will happen. Again, sodium channels open, and then sodium gets into the cell. We go from negative to positive, just about positive 30 millivolts. And that's going to trigger the opening of the potassium gates. The potassium gates will open. All that potassium that works so hard to get in is going to get out. And that's going to take the cell from positive back to negative because all those positive potassium ions are leaving the cell. We call that repolarization. We have that little bit of refractory period or hyperpolarization because so many positive things have gotten out of the cell, but the pump will get us back to that resting potential. Sodium, potassium pump, 3 sodiums out, 2 potassiums in. Good. All right. Here are all those words that we've been talking about. If you want to pause the video and read the words, here you go. Otherwise, let's talk again about that saltatory conduction. All these things that we've been talking about happen only at those nodes of Ranvier, between the Schwann cells and the little gaps of the myelin sheath. Nerve transmission is saltatory. Depolarization, repolarization back to resting potential. Depolarization, repolarization back to resting potential. Only has to happen at those nodes, the nodes of Ranvier or Ranvier. They do not happen under the Schwann cells, under the myelin sheath. So that saltatory jump conduction allows conduction to be crazy fast because we don't have to depolarize, repolarize, depolarize, repolarize every single little micrometer of the whole entire cell. That would make nerve impulses so slow. They're crazy fast because of that jump saltatory conduction. Good. All right. So now let's switch over to this idea of a synapse. We've talked about what's been happening along a nerve cell. Now we're going to talk about what happens between nerve cells. So a synapse is a junction, a gap between nerve cells, or between a nerve cell and effector cell, which is usually going to be a muscle cell. So here we're going to call this the presynaptic nerve cell. This is the postsynaptic nerve cell, and in between those two cells we have the synapse. This picture is helping us see what's happening in this micrograph. So here we've got that nerve cell coming here. Here is the end of one nerve cell, that axon terminal, and then here is the dendrite of the next nerve cell. It is going to collect information given in neurotransmitter form from that the presynaptic cell to the postsynaptic cell, the cell before the synapse, the cell after the synapse. Again, looking at it in this diagram, which is a little bit easier to see than the micrograph, dendrites are going to collect information. We're going to send that signal down the axon to the axon terminal. At the end of the axon terminal, neurotransmitters are going to be released. They're going to diffuse across that synapse. That synapse is the gap between the two cells. Those neurotransmitters, those chemical signals, are going to be transmitted to the next cell. We're going to call that next one the postsynaptic cell, comes after the synapse. This guy was the presynaptic cell, came before the synapse. And then that signal is going to get sent along the cell body, the soma, down the axon, and then it's going to keep going. So quick word of caution about this vocabulary word synapse, the gap between two nerve cells. We have another word that sounds almost the same. Synapsis is when homologous pairs of chromosomes find each other during prophase 1 of meiosis, and crossing over occurs. Remember that these little divots where they're trading chunks, those are called the chiasmata. Crossing over that happens in prophase 1 of meiosis, that's synapsis. A synapse, or plural, synapses, are those gaps between nerve cells. So just be super careful. Don't confuse them, but also remember that this exists. We haven't talked about it in a while, but it is still a thing. Here we're zooming in on that micrograph from a couple pages ago. Again, presynaptic nerve cell. postsynaptic nerve cell. The synapse is that gap between the two cells. Axon, axon terminal, lots of vesicles that are going to hold neurotransmitters here. The dendrite here has lots of receptors, protein receptors, that those neurotransmitters will bind to send the signal along that second cell. Here's that same picture, that same micrograph, but without the help of the diagram. Look at that giant mitochondria, all the energy needed to produce the neurotransmitters, put them into those vesicles, and then the exocytosis of those vesicles into the synapse. Vesicles here in the presynaptic neuron. Postsynaptic neuron are going to have all of these protein receptors that those neurotransmitters can bind to, and the synapse is that gap between those two nerve cells. Here's another micrograph showing the axon terminal of a nerve cell. Can you find it? It's right here. And we're actually looking not at like the nerve cell isn't coming this direction. Instead, the nerve cell is coming like up out of your computer screen. So imagine that the axon goes down into your computer screen. So this is the top of the axon terminal. Again, we've got lots of mitochondria. We've got so many vesicles, and those vesicles are filled with neurotransmitters. Those neurotransmitters during exocytosis will be released into the synapse. The synapse is this gap here. And what's kind of cool about this nerve cell, this axon terminal, is actually communicating with a muscle cell. And I know that because I can see the muscle fibers here. So this nerve impulse is going to tell this muscle to contract. Over here we've got capillary. Notice how thin those capillary walls are so that they can send lots and lots of nutrients to all these nerve cells, because these mitochondria are going to need so much glucose to make lots and lots of ATP, to make all those neurotransmitters and all those vesicles and make all that exocytosis happen. And here are all the details of all the things that happen to get those neurotransmitters out of a presynaptic cell. Here is our presynaptic cell. So they can diffuse across the synapse. Here's our synapse. And bind to the receptors on the postsynaptic cell. Here's the cell that comes after the synapse. So all along the axon here, we've had depolarization, repolarization, back to resting potential. We had those sodium ions, we had those potassium ions, and now we get to the axon terminal, and we're going to throw in a whole new ion, calcium. So now we're going to add in some calcium. That action potential reaches the axon terminal. We're going to open calcium channels, these voltage gated ion channels for calcium. Calcium is going to flow into that presynaptic cell. Calcium is going to enter. It's going to signal these vesicles that are filled with neurotransmitters. These little guys are the neurotransmitters. Those vesicles are going to migrate to the cell membrane, dock with some proteins that sit here. That docking protein is going to help exocytosis occur. Exocytosis is going to occur. Those neurotransmitters are going to diffuse across the synapse and then bind to those receptors on the second cell, the postsynaptic cell. And then we have some kind of signal that is sent along that postsynaptic cell. We're going to open up more ion channels, and it might be sodium, and depolarization could occur. It could also be maybe like a chloride channel, which would actually shut down that second cell, because we do have inhibitory neurotransmitters as well. Speaking of neurotransmitters, here are a bunch of them. Adrenaline and noradrenaline, also known as epinephrine and norepinephrine. These guys are hormones that are released by the adrenal glands that sit on top of our kidneys. This is fight or flight. This is rest and digest. This is calm back down. And then you've probably heard of dopamine and serotonin. These ones are maybe not quite as commonly known. GABA is another calm down neurotransmitter. Acetylcholine is helpful for learning. Glutamate is helpful for memory. Endorphins just make us feel so good. And we're not going to spend a whole lot of time on this, but it's super interesting stuff if you want to do a little bit more research or reading. So imbalances in neurotransmitters can lead to a lot of our mental health disorders. And so again, we're not going to spend lots of time on this, but it's really interesting and super important. So definitely look into it if you're interested. And just like we had before, here are the words that we've been talking about. If you want to pause the video, read the words, make sure that you've got all these details down, you can. Otherwise, I'm going to talk a little bit more about acetylcholine. Acetylcholine is one of those neurotransmitters that is involved in learning, but also in muscle movement. So acetylcholine will be released from that presynaptic cell, diffuse across the synapse, and bind to the postsynaptic cell's receptors. We don't want the acetylcholine stuck to those postsynaptic receptors forever. We want them to move out of the way so that we can send another signal later if we need to move our muscles more later. So what happens is we've got in the synapse this enzyme, acetylcholine esterase. If it ends in ACE, it's probably an enzyme. And it's going to break acetylcholine down into choline and acetate. That's going to modulate or regulate the signal that's coming from the presynaptic cell. Again, we don't want acetylcholine stuck on the receptor forever. We want to be able to send more signals. So acetylcholine esterase, abbreviated here ACHE, is going to take acetylcholine and break it down into two chunks, that acetic acid or acetate piece, and then the choline piece. And those pieces will be taken back up by the presynaptic cell or some other cell and then turned into more stuff that we need. We have these pesticides known as neonicotinoid pesticides. They look like the molecule nicotine, which is why we call them neonicotinoid pesticides. And what they do is they have a shape that is super, super similar to acetylcholine. So what should happen is acetylcholine diffuses across the synapse, binds to the receptor, acetylcholine esterase comes in, breaks it apart, it gets pulled back into the cell and gets turned back into more acetylcholine most likely. The postsynaptic cell gets some acetylcholine and then it goes away, and then it can get more acetylcholine and then it goes away. If we have these neonicotinoid pesticides in the body, what happens is those pesticide molecules, because they look very similar to acetylcholine, they're going to bind to the receptors on the postsynaptic cell. Acetylcholine esterase has no effect on those neonicotinoid molecules, which means that all this acetylcholine being released by the nerve cell has nothing to bind to. And so there is no impulse sent from this cell to that cell. It's blocked. And that blockage is going to paralyze the bugs that eat the pesticide that's on the flowers, because they cannot send any signals from their neurons to their to their muscle cells. No signals to the muscle cells means you can't move your muscles, means you're paralyzed, and then they die. And that's great if you want to save your roses from aphids. It's not so great if you're a bumblebee and you just want to make some honey. We actually have some big concerns about neonicotinoid pesticides in our environment and the numbers of bees that are declining. So this is a version of neonicotinoid. These are our bee colonies, and you can see that they are definitely in decline. You might think that bees are not that big a deal. We have other things other than honey, so do we even need the bees? If we don't have bees pollinating plants, then the plants don't make more babies. And then we don't have plants, and that should worry us a lot. And also, bees are cool. If you want to pause the video and reread all about neonicotinoid pesticides and acetylcholine, here you go. Otherwise, my friends, we're done. Objectives achieved. We talked about the structure of neurons. We outlined saltatory conduction. It jumps, jumps, jumps. We talked about propagation of resting potential. 3 sodiums out and 2 potassiums in. That's going to lead to a negative 70 millivolt. We talked about those stages of action potential. The depolarization when the sodium gets in, the repolarization when the potassium gets out. That little bit of hyperpolarization while we wait for everything to get back to normal. We annotated those action potential graphs. Resting potential, depolarization, repolarization, hyperpolarization, back to resting potential. We defined a synapse, that gap between 2 nerve cells. We talked about the stages of neurotransmission. We're going to have some calcium channels open. Vesicles are stimulated. They go through exocytosis. Neurotransmitters diffuse across the synapse and bind to receptors on the postsynaptic cell. We talked about those pesky neonicotinoid pesticides and neurotransmission and how they paralyze bugs. You did it.